Okay, all right, folks. Good evening. I'm Jorge Bogantes with the Anacostia Watershed Society, and welcome to our Watershed Wednesday Happy Hour. These were normally held in restaurants or bars in DC, with folks giving interesting presentation about different topics, but because of the pandemic, it's been in Zoom for a while, for a year or so. And tonight we have a very special guest. Her name is Anacostia, and she's a co-founder of Capital Nature, is a local nonprofit based in DC. So Anna is a certified forest therapy guide with the Association of Nature Forest Therapy Guides and Program. She's also a master naturalist from Virginia. And she's committed to connecting people with nature through her organization, Capital Nature. And Anna is originally from Hawaii. She has been in the DC area since 1994, right?
96,
yeah. And she's an experienced tour guide and sustainability educator with 18 years of experience in the field. She's also in the leadership team for the DC area City Nature Challenge, which happens every spring, international event, and on the steering committee for the Washington Area Citizen Science Network. So Anna is an avid bird watcher and nature photographer, and she loves to do interesting hikes all over the DMV area, looking for different things, whether it's otters or mushrooms and whatnot. And we're very excited to have you tonight, Anna.
Thank you so much, Jorge. Thanks. I didn't have to read my slide here, so that was awesome. That saved me one slide. That was great. Welcome, everybody. And I put my name there phonetically. You know, Anacostia, Jorge, you did a great job with that. Sometimes it takes a while for people to get my name. I am a nature nerd, sort of by heart. And so those of you on the call that know me know that I get really excited when I talk about citizen science and iNaturalist in particular. I don't work for them. I'm just a junkie, and so I like to spread the good word about using iNaturalist. Once, as a photographer, once I learned that my photos that I took can contribute to scientific research, I got really excited about that, and so I wanted to kind of spread that with everybody. So before I get started, I just want to tell you really quickly about my organization. It's called Capital Nature. You can visit the website capitalnature.org. You know, we're just committed to getting people connected to the nature in the region. There's so much of it, so we gather a lot of information. We put out these little monthly newsletters about things going on in the region. One of our biggest features on our website right now, we're updating with a new webmaster this week, is our calendar. And so we have been curating a calendar for a couple of years now of all these different events going on, both virtual and in person during the pandemic. So that's being updated right now. So there's nothing on there right now, which I'm embarrassed to say, but check back in a week or two. We will have populated our calendars moving forward. And so we do nature and tree walks. I do forest bathing and virtual forest bathing for folks, but we're super jazzed about citizen science. So that's what we're going to talk about tonight. So what is it? What is citizen science? Some of you on the call may know, but if you don't know, it's also sometimes known as community science. So when people get together and help scientists gather data, basically scientists can't gather all the data for all the projects that they're working on. So if they have people out in the field that can gather stuff for them to help towards their research, it gets things done a lot quicker. And what's great about citizen science is anyone can do it. What do? You volunteer your time, basically. You go out and observe, you collect and share data, and you submit photos and other evidence of things that you find. Why is it important? Well, we really need that nature connection, especially now more than ever. Again, you're contributing to science, and really you're supporting your community and the environment that you live in. So what's a BioBlitz? That's why we're here. That's why we're learning about iNaturalist, because of the Anacostia River BioBlitz, and it's intensive survey of a defined area. So we can inventory as many things as possible in a short amount of time. Sometimes it's a 24-hour BioBlitz. Sometimes in this case, we're Thursday, Friday, Saturday, we're a four-day BioBlitz. Other times it's a week, other times it's a month. So depending on who you participate, whatever BioBlitz you participate in it, that time is definitely defined. Why should we have it? Well, it's a census of nature, so it helps us create a list. So for example, Jorge and the folks at Anacostia Watershed Society, they would really like to know what species are in their area. And so citizen scientists are helping to gather that information. Also, it helps land managers make decisions. For example, a good example, our friend Alonso Abogadas, the Capital Naturalist, he uses the example of people are using iNaturalist and reporting all these invasive species on different Arlington parks. That informs their team in Arlington that, oh, perhaps we need to go over and do an invasive pull in this particular area because of the data that people have submitted. So it's helping people make decisions about the land. So what's iNaturalist? It's an app. It's a free app. It has both a phone or device interface, and it has a very robust online on computer interface, which we will walk through. And so basically, it's a place where you can record things that you've seen. You're comparing your information with nature lovers, and you're learning about the different species that you observe, and all of this gets contributing to science, and I'll get into the details on how you do that. So I imagine people on this call or on the Zoom like to take pictures using their phone. I mean, we all like to take nature pictures. And what's great is our camera pretty much, it's so convenient to just kind of pull out your camera or your phone and just take pictures of stuff. And in most cases, if we let it, it will be geotagged. So it will mark the day, not only the day and time, but it also the location. And that's what's really helpful for scientists. So the first thing you need to do, if you haven't done so already, is create an iNaturalist account. So just by going to the website or putting the app on your phone, which is available in the App Store or Google Play, go ahead and sign in and start making observations. And there's all kinds of videos, and we'll talk about a couple different places where you can find videos, including the Anacostia Watershed Society website. They have links to videos on their BioBlitz page as well. And what's great about it is it works across platforms. And so, if you have your iPad, if you have your phones, and again, like I mentioned, using the desktop, there's just, it's all seamlessly connected. But I will say that the desktop interface lets you do so much more. So these are the 5 major characteristics of what making an observation in iNaturalist is. It's recording who you are as the observer, what you saw, when you saw it, where you saw it, and evidence of what you saw. And usually that evidence is a photograph, but it can also be other things like sounds. And so it records an encounter, a one-time encounter between you and an organism. And the organism doesn't have to be living necessarily, and we'll get to that in a second. It could be evidence of life. But here I have seen this eastern painted turtle over at Huntley Meadows in Alexandria. If I were to walk away and come back an hour later and take another picture of this possible same turtle, it's a separate observation. It's a distinct point in time with your interaction with that being or evidence of life. So making observations, how do you do that? So if you have your phone, and I will say that the interface looks a little bit different between iPhones and Android, but they all do the same thing. They just might have a different name on their button. But basically you would start by taking a photo. And the first thing it's going to ask you is like, do you like this photo or do you want to retake it? If you happen to take a really blurry photo, then yes, retake it. That's the one thing we want to say is no blurry photos in iNaturalist, please, because that really doesn't help anybody if you put some blurry photos in there, because no one's going to be able to help you identify it. So you take your picture. And if you notice on the second panel here, there's a plus sign. You can take more than one picture, and we encourage you to do that. Take a picture of the actual item or specimen, but maybe take a picture of it close up. Maybe take a picture of it further back to show it in context. Is it at the base of a tree? Is it coming out of the water? Is it on a mountaintop? And so all of these things are going to help people with identification.
Oh, I need lots of cheese.
If you look on the second panel here, you'll see that the date and time have been captured. The location's been captured. The geoprivacy, which I have a slide for that a little bit later, but basically this is where you set if this is open for people to see the location or if it's private. So for example, if this were an observation that was made in my backyard and I didn't want everyone on iNaturalist to see where my backyard is pinpointed on a map, I could obscure that. And I'll show you a little bit of slide for that later. But this is where you do it in this actual, when you're making the observation. Captive cultivated, it's got to be wild. So we're looking for wild things. We're not looking for your pet cat. We're not looking for the roses that you have planted in your garden. iNaturalist is solely focused on wild things. So after you've taken your picture and you've gotten some of this data, you go ahead and click this, what did you see? And what's great about iNaturalist is it has this huge database that's going to give you the top 10 guesses if it can, based on your photo, if it's a decent photo, of what it thinks it might be. And sometimes it might just be able to get it to the genus and not to the species, but anything is good, because if you just put nothing in there, no one's going to be able to like find your observation to help you identify it if there's just nothing in there. So we want to pick something. So once you, let's say you think you're not sure which kind of daisy or fleabane this is, maybe you just pick this particular genus and you click share, and then you've uploaded your observation to iNaturalist for everyone to see. So how does it work? Right now, the last time I talked to our friend Carrie Seltzer, who works for iNaturalist, she said there were 90 million photos in the database that are being analyzed across all of our pictures. And so right now, they're actually only using a third of those. They're actually only, they haven't like processed everything. So right now, our pictures are getting compared to 30 million photos. They're going through and trying to sift to kind of see what, and so it trains it to be artificial intelligence so that it can make some suggestions for you. So here's an example. So there's this picture of this violet, and yep, it's definitely, they say it's definitely in this genus, violets. But as a lot of us know, there's several different types of violets and they all look really similar. So some ways of getting around these is clicking on each one and looking at, say, where these violets are commonly seen. If you pull up a violet and this one is only in Africa, then you don't want to pick that one. You want to pick one that might be in our region, the ones that have been cited in our region. So they give you all these options for you to click on so that you can compare your picture to the pictures in the database to kind of see if there's a match. So the things that have to be part of an observation, it definitely has to have a date. It definitely has to be geo-referenced and have coordinates. It definitely has to have evidence, which is mostly photos, but sometimes sound. And like I said, it can't be captive or cultivated. It can't be your pet or something domestic or something that you've planted in your garden. And so if something doesn't have that, it's considered needs ID. And that's, needs ID is where we all come in as helping each other in iNaturalist, is helping each other to make these identifications because we want to get it to research grade. So we'll talk about research grade. So research grade is where the community, at least 2 out of 3 people agree that yes, this is a barn swallow, or yes, this is a red-headed woodpecker. So if I made this observation and I said this was a barn swallow and someone else came up after me and said, no, Anna, I think this is a cliff swallow. Okay, so then we have a little debate going on. So then I have to decide, like, do I think it's a barn swallow or a cliff swallow? Other people could chime in. One of the things that I do, and we'll see if we can show you this when we go on to the actual site, but you can actually ping experts in iNaturalist. Like I can look and see who has made the most observations of barn swallows in iNaturalist, and I can message them in iNaturalist and say, hey, Joe Barn Swallow, do you think this is a barn swallow? And he'll let me know. And so you're basically the total strangers all over the world that are experts on these different taxa are able to help you make these identifications. So as of today, just did a screenshot in iNaturalist, over 80 million observations, 346,000 species, ID'd by 216,000 people, 1.8 million observers around the world. So you would be amazed. I remember the first time I made an observation of a bald eagle, somebody in Russia, like 2 seconds after I posted it, verified, yep, this is a bald eagle. So there's somebody on the other side of the planet that's looking at something that I just took a picture of. And that just kind of blows my mind. So let's talk about the BioBlitz, the Anacostia River BioBlitz. As of today, this is a screenshot of the cover page of the BioBlitz page in iNaturalist, and you'll see the link up at the URL at the top there. And so we're going to ask you to we want to ask people to join the project. And there's a little thing up here that says join or leave up in the upper right-hand corner. And that because the organizers such as Jorge and their team, they're able to kind of see who's actively participating in the BioBlitz. Now, but if you happen to be making observations within the geographic area that is bound for the Anacostia watershed, if you make it during those 4 days that we talked about, September 16th to the 19th, they'll be counted regardless if you join the project. They'll be counted anyway. So, but we do encourage people, like I said, to join it anyway, just because we can actually see how many people are participating. So right now you see the BioBlitz, which covers several years now. This is the 5th annual, 164,000 observations of almost 7,000 species, ID'd by almost 6,000 people, observed by almost 8,000 people. So that's a lot of people that are making observations in the watershed. Jorge gave me this great little list here, a little graph here to kind of show you sort of the last couple of years of how things have been going in the watershed and for the BioBlitz. And so I love it because it says like what the weather was like during that period of time, because it's not the same dates every year. Obviously they pick it, based on schedules and stuff like that. And so things might have bloomed or not bloomed. Things may have been migrated or not migrated by the time we've been doing this. So it's interesting to see the differences and how many people have been engaged. Now, Jorge, I see that you have a 0 here for last year, but I know that there were people that did this last year, correct, Jorge?
No, that's actually, yeah, for the in-person, we didn't have anything.
Got it. Okay,
Yeah.
So for the in-person, but for the virtual, people definitely.
Actually, for the virtual, I don't know. Okay, if you look at observers, you actually are going to see that the number has been growing in the last.
Got it.
So,
I see that there. Yeah, thank you for making that distinction between the live event versus the people that are doing it virtually. So what I love to see is I love fungi, I love mushrooms. And so seeing the jump between 2019 and 2020, look at that huge jump from 23 to 116, like that's super exciting to see that, people are, paying attention to these different fun taxa. And then when we do dive in on the back end, one of the things that we'll see is that there's a leaderboard. So we can see who's making the most observations, who is observing the most species, and what are the most common observations, the top observations. So right now it's the common slider. So common slider, American robin, eastern gray squirrel. So no real surprises here, really. Okay, so I'm going to jump out of this and I'm going to go over to the website. Can you see that? The website? Yeah, okay. So here we are on iNaturalist, and get rid of this thing. Again, here's where you leave or join up here. So when you navigate to the site, you'll just we ask that you join it. The dates gives us some information about the BioBlitz, the stats that we just looked at, and you can actually, so you can click now. So let's click on species. And now we can see sort of in a grid view of all the species, the top species, and how many observations have been made of each of these things. So you'll want to look at these, and so that just kind of tells you as a preview before you go out and participate, these are the types of things that you're probably going to see because these are the most common. So looking at species, looking at observations, I wanted to point this out here. Here we go, observations, yep. So and it just kind of shows you who did it, when they did it. You can click on them and see like where, like if you're really curious on where somebody saw this great pond hawk, you can click on it and there will be a map that you can zoom in on and look exactly of like where the person saw this in the watershed. So you can just kind of poke around to kind of see all the different things that have been already collected. The other thing I wanted to mention is, I told you there are some robust tools on here, but one of the cool things is the community of users. And so if you go up to community here and click on people, you can search for people. So for example, if you're out at a park this weekend and you meet, Mary Ford from National Geographic, you're just like, well, I want to follow her now on iNaturalist because I think she made some really cool observations. So we can click people. And then it's going to give us a little search bar here where I'm just going to search for Jorge and search. And now I have 2 little things that come up for Jorge. So I'm going to click on this top one here because I think that's Jorge. And so now I'm looking at Jorge's sort of personal iNaturalist page. Up here, I'm actually following him. It says stop following him, but there are some of my friends, my nature nerd friends on iNaturalist, I follow them. You could follow, let's say you're like totally into mushrooms. You could look and see who the top mushroom people are in the region. You could follow them. You could message these people. Like I said, if you want to get some help on some identifications, we can look at Jorge's observations, which he's been making all kinds of observations because he's been out in the fields doing his mussel surveys and finding all kinds of things. So you can kind of look and see what people have been observing, and you can tell that some people have certain things that they really like. They really like taking pictures of. Let's see what else. Projects. So if projects is a way to, you can go up and look up projects. And let's say you're into here. Let's try fungi. Here, I keep mentioning fungi. So if I click on fungi, these are all the different projects in iNaturalist, like plants and fungi of the world. Well, heck, that's everything, right? So maybe I should join that one. North American mushrooms and fungi. So these are ways that you can join as many projects as you want, so that if any of your observations meet the criteria for those projects, they're going to show up. They're going to show up in all these different projects. Okay, so I'm going to go ahead and get out of this and go back and show you guys. I'm gonna ask Jorge. We pulled up a sample observation from last year's BioBlitz. And Jorge, do you just want to tell us a little bit about this friend here?
Yeah, this was my favorite observation from last year BioBlitz. It's a ball jumping spider. I've been fascinated by these little critters. And I found this one at my house. It was actually inside, and it was in the living room. So we put him out on the front, and I got that nice shot. I was using one of those lenses, like the one Anna showed in one of the pictures, the one that you can clip on the smartphone lens. And yeah, I mean, these spiders are very unique because they are great sneaky predators. They sometimes have predators that is comparable to cats, so they're pretty fascinating. And we have around 50 species in the region. So yeah. Of these.
And what's a great example to show here is that when Jorge made this observation, he made a guess at the genus. Someone named Jumping Arachnid suggested the bold jumping spider. Jorge saw that and he agreed with that. And so the fact that these two agree on that assessment, and notice he's got a couple of different pictures in here, that got us to the research grade for that. Now, if another person, let's say another person came up and suggested a different type of arachnid, then, Jorge would have to decide, and so we'll go back and forth until there was sort of a consensus so that we could get to research grade. So this was just a great example. So thank you so much for showing us that, Jorge.
And then if someone decides it is, another jumping spider, like a green jumping spider, then it's gonna show as Maverick, right? Because it's like, going against the grain,
yeah.
sort of thing.
Yes, yes.
Yeah.
To try to like convince you otherwise. Yeah. I also wanted to mention that if you see down here in the project, this particular observation is automatically in this biodiversity of the Anacostia River, which is what we're all contributing to. But he has also joined the Maryland Biodiversity Project, which if those of you plan on making observations in the state of Maryland, I highly recommend that you join that project too, because everything goes into that project. And I'll show you an example of how that comes into play a little bit later. Let's see.
And they have an amazing database of pictures of all kinds of organisms in Maryland. So make sure to check it out and Google Maryland Biodiversity Project.
Absolutely. So what to observe? I start with things that you love. I love, as I said, mushrooms. I love butterflies. I love flowers, things that I'm looking at anyway. Why don't I like to learn more about them. What are you noticing? So I mentioned before that, I am a certified forest therapy guide, which means we do these meditative kind of Zen nature walks in nature where you connect to nature with all your senses. It's not a naturalist walk. It's kind of like just connecting to nature with your senses. And the biggest proponent of forest bathing is slowing down and observing. And so the big question that we're asking ourselves is, what are you noticing? And so when you're walking very slowly through the forest, you might notice little buds like this. And so in the springtime, I go through these periods where I'm sort of obsessed with all these little baby leaves that are coming out. And so those are things that I happen to notice so that I start taking pictures of. Some things are really easy to identify by iNaturalist just by virtue of maybe how unique they are shaped or look. For example, these tulip tree leaves have this very unique, I like to say they're kind of cat face leaves, so pretty distinct, so that when you take a picture of just one of these leaves, iNaturalist is gonna pretty much 100% give that to you as a tulip tree. However, if you took a picture of just of an oak leaf, there's so many different types of oaks in the area that it might need a little more information. Like you might need several photos for it to kind of narrow it down. Same with this sweet gumball here. This is from a sweet gum tree. This is the little seed, a pod that has usually about 60 little seeds inside that kind of disperse in the wind. The only other thing really in iNaturalist that kind of looks like this is maybe a sea urchin. And so this is like when I'm showing people or doing demonstrations live, showing how iNaturalist works, I usually have one of these in my camera bag because it's really easy just to kind of show people because it's very easily identifiable. And some things are easy to photograph, meaning that they don't walk away from you. So they stay still. You know, we have two different things here that this one on the right is actually my favorite iNaturalist observation to date. It was from the City Nature Challenge a couple years ago. Once I learned what it was, I was super excited. And basically it's a zombie fly. It's a fungus that has gone inside a fly, made it go to the end of this stalk of this seed here and grab onto it. And it basically, you know, just took it over. Basically it controlled its brain and then it kind of grew inside it and all that. It's really gory and disgusting, but to me, I was so excited because I was taking a picture of this and I'm like, why isn't this fly moving? Wow, this fly is just kind of hanging out. Let me take his picture. Well, I didn't know he was a zombie. So anyway, that was a pretty, and then we have the Virginia bluebells on the left. Some things are not so easy to take a picture of. If you like dragonflies or damselflies or even birds, you know, they don't sit still for very long. But if you give yourself some time and some patience and get to know the behaviors of these different species. For example, I've learned, you know, with dragonflies that they like to perch like this. Some like to perch going kind of straight out. Some of them like to perch going up and down, which is called obelisking. This is kind of a way for them to kind of chill out, dry out, you know, get their strength back. They usually perch, and then if you kind of approach, they'll fly away, but then they usually come back to the same perch within maybe 30 seconds. So if you just see one and you see it fly away, if you just stand still, there's a good chance that it's going to come back to where you are. So these are the types of things that once you notice the behaviors of these species, then you might have a better chance of getting pictures of them. So we talked a little bit earlier about wild versus captive cultivated. We want wild things in iNaturalist. This is my dog, Lonnie. She thinks she's wild, but she is not. And so I do not document her in iNaturalist. Other things that we find in, say, botanical gardens and things like that, these are cultivated. These are things that are grown by others, by humans. So these are things that we are not looking for in iNaturalist. And I love all these pictures. And I want to acknowledge one of our colleagues, Katja Schultz at the Smithsonian Natural History Museum. She is an amazing nature photographer and a bug specialist. She knows Rock Creek Park like the back of her hand. She and I have presented together on photography for iNaturalist. And so she's given me the permission to use a lot of her photos. And so these are some of them. Signs and remnants, any evidence of life. So we talked about that turtle earlier. Yep, that was a live turtle in Huntley Meadows. But any evidence of life also counts if it's identifiable. And so if this skull is identifiable, this snake skin, this piece of a butterfly, this feather, this den, these footprints, the scat and the poop, the frass, if any of those things are identifiable, that's fair game. So if you see something like that, a nest, you know, a lot of them have characteristics that there's someone in iNaturalist that can help you identify that.
I have a question.
Sure.
Regarding your previous slide, so things that are cultivated, what about, so like a lot of invasive species particularly are things that are, you know, ornamental plants that have then escaped to the forest. So say if I find a wisteria plant climbing in the forest, does that count as wild?
If you think that's a what do you call it, a pioneer, or like something that's gone off on its own, and you don't think it's planted by a person, yeah, you could definitely do that. Yeah.
So like weedy species generally in say a cultivated crop field that has adapted to the human influenced ecosystem, that's fine too as well.
Yeah. Thank you for asking.
I would say when you're in doubt, if it's like on a public space, maybe you can put as a common, possibly cultivated, not sure. Yeah.
That's a great point. I didn't mention that before. Thank you. There is a comments area when you're making your observations. And so any additional information, especially if you're unsure about something, putting it in there saying, yeah, I found this in X field with X kind of plants or that type of thing, that will kind of help people make the assessment. Definitely. Thank you, Jorge, for showing that. Thanks for that question.
I find something very pretty in the environment and realize, oh, it's an ornamental plant that has escaped.
Yeah. Thank you. Audio. So audio sounds are also evidence of recording in iNaturalist. They now have a way for you to do that. And an example that I like to use are the crows. We have two different types of crows in this region. We have the fish crow and the American crow. And so they both they look very similar. And so really the best way to distinguish them is by their sound. So, an American crow would be sort of a caw, caw, caw sound. And a fish crow is more of a nasally sound, kind of like a nuh-uh sound. So when you hear the difference, you could upload that. And usually, especially the bird nerds, like myself, making identifications, hearing that sound would help make an identification. Tips for observing. Get close, especially when you got little things like this, insects and such. And Jorge did mention you get very inexpensive little clip-on macro lenses. They range in price. You can get really cheap ones, you can get really expensive ones, it just depends on what your budget is. Clip those on your phone and you can get some really cool close shots. A lot of our phones these days, you can put it in macro modes and things like that. So it's amazing what our phones can capture, but getting close, getting context is great. So showing that this is on some bark of a possibly down, looks like a piece of bark next to a mushroom, and then getting close. Okay. And sometimes I know Katya says when she's talking about her insect photography that it's really hard to get all the different parts of, like, say, an insect in focus in one picture. So take one from the back, take one from the front, take one from the side, take one from the top. So if you get all these different angles, that's going to help paint the picture of what the species is. And focus. I mentioned that at the beginning. Please do not upload any blurry photos. It's just taking up space in the database. No one's going to be able to, I won't say no one, so maybe some experts might be able to look at this on the left and be able to identify, but really try to, if you have a chance, when it asks you to retake that photo, do you like that photo? Yes, retake it if you're in doubt. Select your focus. So, you don't want to just walk into a forest and hold your camera up and just take a picture, because that could be, it could be a tree, it could be a bird, it could be a bug, it could be a mushroom, it could be anything. And so not sort of narrowing in on what you want the identifier to look at, that makes it really hard. So just definitely select your focus. And then even separate your subject if possible. So, if you have a way of blurring the background, or sometimes I carry around like a piece of cardboard or a piece of white paper, especially when I'm doing like trees, like with a lot of needles and fir needles and stuff like that, there's just a mass of needles. But if you put a piece of paper or something behind it when you take the photo, it's going to let you kind of zoom in on the features of just a couple needles and not the whole thing. Details are really important. What color are the eyes of the dragonfly? What are the features on its abdomen? Are there any markings on its wings? This snapping turtle that Katya got, which is amazing, just kind of, all the different facial features, like all these are going to help people the more detail you get. And then different perspectives. Again, if you can get down to the level of your species or what you're looking at, get those angles that are closer rather than just kind of like shooting down. And I'll show you an example of that in just a second. And also look for things on plants and other things. So for example, this tree here was actually planted at a library near my home. So this is not something that I would put on iNaturalist. However, this ladybug on it is fair game. Same with these maple leaves here. The maple tree was planted in the parking lot, but there's maple tar fungus on these leaves, and this particular fungus is something that I can report on iNaturalist. So look on things. Working with others is great. I am birding with my godson and his brother. So if you're out with other people, you can help each other find stuff, because it was something that you might see so clearly, might be missed by others, and you can just kind of help each other out. So some of my favorite things to observe: trees. I love trees and shrubs. And so when you're doing a tree observation, these are the recommendations that we make. You want to get a picture of the leaf, showing the leaf and how it's attached down here at the petiole to the branch, and to show you the venation, to show the shape of the leaf, whether or not it's pointy like this or if it's rounded. Take pictures of the bark of the tree. Sometimes scale, like if sometimes when we take pictures of bark, you might want to hold up like something like a quarter or a dime, so that you can kind of get the perspective of like how big these ridges are. If there's any fruit or nuts. And then sort of a backup, take a picture of the tree, so show the shape, the overall shape of the tree. And all of these things, again, are going to be the clues that people need to make identifications. Same with mushrooms. Now I mentioned taking pictures down. So a lot of people, when they take a picture of a mushroom, they're just like, oh, there's a cute mushroom in the grass. I'm going to take a picture from the top down. And so that's a top-down picture. But you must get pictures underneath the mushroom. We need to see if there are gills here or spores or what's going on under the hood. I usually carry around a little mirror like this that I can flash up there and take the picture so I can get the bottom of it. How it's connected to wherever it's planted, the stem or the stipe, all of these things help. And just showing people like this is on a rocky hill, or this is in the middle of some grass in a yard. All of these tell us a little bit more about where these things are so we can ID them. I love butterflies, and as we know, butterflies have different colors and shapes based on how they're positioned. So if you're looking at them from above, you can see the top part of there, just the different little dots and features, but also from the side, they might have a different look to them. So it's good to get multiple angles. Dragonflies and damselflies, I mentioned, here's one obelisking here that's kind of going up high, that's kind of holding its position there. What are the patterns on the wings and on the body, and there's stripes on it, and what are they doing? So over here, these are damselflies, either they're about to mate or they've just mated. So where the male is holding the female by the neck, and they usually make a circle or a heart, and that's like sort of their mating thing. So these are things that you'll see and get details of. Birds, I also love, I'm a birder, so just love taking pictures of birds, seeing them in their environment. When you have the male and female that have they're sexually dimorphic, where the male looks different than the female, or like say in the cardinals, those are really easy to identify pretty much when you're taking the pictures. Couple tips and reminders. If you're out and about, make sure that you don't want to trample on stuff. And if you've moved a log, perhaps to look underneath it for a salamander, make sure that you put things back gently so that you don't disturb all these microhabitats. Really good to bring a phone charger when you're out there, because you have the chance of like using up a lot of your juice while you're out there. I mentioned using a coin for scale, or sometimes people have small rulers, using the mirror like we saw. There's hand lenses, also called loops, so if you want to see really closely. Sometimes if it's a poorly lit day and you want to get a better light on your subject, grab a little flashlight, or even just use the flashlight on your phone. And as I mentioned, using paper or cardboard behind your subject. Another pro tip is when you're in the settings on your phone, you can set it. Some people say like, oh, I don't want to use my data. Totally fine. You can go and uncheck automatic upload, so that you're going to make all your observations and it's not going to upload until you go and join a Wi-Fi or join a network, so that you don't have to use up all your data while you're out in the field. Geoprivacy, so we mentioned this a little bit earlier. As you're making your observation, there's on the observation, there's a thing that says geoprivacy, and so you can set it to open, obscured, or private. Now, for the purposes of what we're doing for the Anacostia BioBlitz, things need to be open so that people can see where you made the observation. If all of your observations are private because they're in your backyard and people can't see where they are, we're not going to be able to get it to research grade because we don't know where it is and we can't make an identification. So for the purposes of this BioBlitz, we ask that you keep things open. And if you need help, they have great, their website's great. They have a support button where they're very helpful, where if you run into a glitch, a technical glitch of any way, you can actually email them. They have a really great forum where you can just pop things in as topics in a forum, and there's all kinds of little conversations that you can learn from. And then you can also ask other users. So I mentioned that you can go in there and tag other people. So if I were making an observation, say of a mussel, and I didn't know what kind of mussel it was, I would probably go in there and I would tag Jorge. And you can actually only do this, I believe, on the website, not on your phone. And tag Jorge and I can say, Jorge, is this X kind of, is it alewife? Is it alewife? Is that a kind? Jorge?
Yeah.
Yeah. And then you could help me out with that.
alewife floater. Yeah.
alewife floater. Okay, I was close. That's close. Let's see.
And also you can do alerts, right, for taxa. Let's say you're interested in asters. Maybe
Yes.
you can put that that's on the desktop. That's not available.
Yes, and actually here, let me just jump out and show that to you really quick. Because let's see, your observations. Okay, here's me.
Yeah, I do that for mussels, and I get the updates and from all over the US and sometimes actually the world, because freshwater mussels are kind of rather rare. So
under my profile, I have followed, it looks like the fungi, including lichens. I've just picked the geographic area of Washington metropolitan area to see what comes up. And then I guess at some point I did maple leaves, like maple trees. So you can pick, if you're totally into a certain taxa, you can go and follow these different kinds. And that's really helpful. And then these things like literally just pop up in your personal feed. So yeah, thank you for mentioning that. Oh, and actually while I'm in here, let me just go into an observation. Let's take this one. So this in this comment field is where, I was actually with Saranella Leaderboard, one of our great friends and mycologist from the Audubon Natural Society. If I wasn't sure about this, I would just at Saranella and say, am I right? And then she would see that in her inbox up here. There's like messages, and so then she would be able to say yes or no if I was right on this or not. Also up here, this little way up here, this tiny little what do you call it? A bubble. These are all the different. Oh, cool. These are all the different people that have made added identifications or comments to my personal observations. And there's a bunch of rock stars in here. And one of them I just want to point out is Luke Reptile, is a teenager in Virginia who's a herpetology expert. And so if you have anything that's like snakes, turtles, salamanders, he's a great person. I tag him on my herp stuff all the time. So you'll get to know. And here, when I scroll down here in the bottom right, you're going to see the top identifiers of this particular species that I'm looking at. And so I could at David Enrique in here and say, David, do you agree with this? And then if David doesn't respond, maybe David doesn't check his notices, maybe I'll go down to the next person, John, John, do you do, so again, it's a social network, and you end up meeting and making friends with all these people around the world based on your observations. Okay, how are we doing on time, Jorge?
Yeah, it's 7:19. Yeah, we still have time.
Okay, great. Okay, so we did that. Okay, what happens with the data? You're probably curious, like, oh, my data just goes into iNaturalist. Well, it actually goes here, the Global Biodiversity Information Facility, or GBIF. So basically it's this clearinghouse online of all these different data points from all these different organizations, so that researchers, students, they can go and kind of search this database for different data sets, to work on their reports and assessments, and the projects that they're working on. So we're basically all putting it into this sort of collective area. Millions of data points, which is all helping, especially towards conservation, which is wonderful. So here's an example of looking at these data points. You know, phenology is looking at the timing of biological events, and what they're noticing after all of these years of making observations, you know, are things blooming earlier? Are birds not being sighted as further south as they used to be because of climate change and that type of thing? So all of these things, when plotted together, are helping, you know, helping us understand the biodiversity and what's going on with them. And you know, I had made this observation of bittersweet nightshade and was contacted by the folks that put together the something called the Socrates Project, which is the poisonous plants in Virginia. It's a book. And so they went through iNaturalist to look for specific plants. And then they actually contacted people like me and said, hey, can we put this in our book? And I'm like, well, heck yeah. You know, so something that you take might be added to a project. Are there any questions? I have a few things after this, but does anybody have any questions in the chat or anything? Doesn't.
Yeah, the only question, Lisa Garrett, she asked, are there assigned areas to blitz? Are there taxa groups to join each day, or is it all on your own? So we will have events every day starting tomorrow, for example, at Buzzer Point, 10 to 1. We'll do an aquatic survey, and then there's be different events. The ones for Friday and Thursday and Friday have limited slots. The ones on the weekend are more open, especially the one at Kenilworth. It's a mix of pre-registration, but also people that are walking in the gardens can join us and learn. We will have experts also from the Smithsonian Environmental Research Center, experts in aquatic biodiversity, and mushroom experts, plant experts, and whatnot. So yes. Yeah. Oh, there's a new message. Oh yeah, Lisa, where do we see these slots? You have to go to the link I provided and click on each event. It'll take you to the registration page, and that's
here I will. I'll jump out really quick and go to that. Okay, so here we are on the Anacostia Watershed Society webpage, and here's the BioBlitz page. And so it goes through all the different events that have been scheduled and how you can register for them. Mm-hmm. So I will put this link in. Is that link in the chat already?
Yeah, it's already in the chat.
Got it,
yep.
got it.
Yep.
Got it, yeah, okay, great. So I'm gonna go back. Okay, so Jorge, there's Jorge there, yay. At one of our previous ones. So what are we doing this week? We're exploring the national treasures of the river, or you can just be in your backyard. So it's wherever you want to go. When we're documenting the biodiversity, we're using the app anywhere in the watershed. And then we're just learning about all the different species that are around us, which is super exciting. Why are we doing this? Because we all need to be outdoors. It's a cool app and you'll be cool if you use it, in my opinion. You'll learn about all the stuff that we share our world with, gather information, and you expand your social network. And so you can do things, again, individually, just go out on your own, or be part of a safely distanced group, like some of the events that we just showed you on their page, which is this. This is just from the flyer, and just know all the different organizations that are participating in some way.
Sorry, Anna, there's a question by Sofia Leonova. How should we deal with observations of feral or domesticated but free-roaming organisms like cats?
Feral cats.
There's actually a project in.
There is a feral cat project.
Yeah, they use trail cameras, and yeah, you can take pictures of those, and maybe I don't know if they are collected immediately by that project, or you can sort of tag them to that project.
You can also do a project in the project. I know there's a feral cat project. Mm-hmm. I just haven't participated in, I don't see those, but yeah, and then your observations will get added to that.
Yeah, if you're sure or that the animal is, you know, feral, or if not, you can again put in the comment possibly feral. But that's a good observation because yeah, cats have become a big issue. You know, they kill a lot of birds and small vertebrates. So yeah, that's an observation of scientific interest.
That's a great, and it's a great thing to know, because if lots of people are making observations of feral cats in say X area of the city, I mean, that's information that's good to know. So that's a great question. Thank you.
Actually, last year I was looking at the project data for 2020, and the most observed species was cats. And I was, what is that? And then I went, and it was all because of that feral cat project. And I was, okay, that's great.
Interesting. 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 Yeah. Wow. Because I know it is a problem. Yeah. Just really quickly, there are toolkit, like if you're working with, if you're an educator, you're a parent, you're taking care of kids. And actually this is about the City Nature Challenge, but it still comes into play. They've put together this education toolkit on the City Nature Challenge site, which is this major global BioBlitz that a bunch of us participate in every year. And so I just wanted to point that out because it's got like tools, like if you want to use iNaturalist in your classroom or take your kids out on the weekend, there's things to do there. So that's kind of helpful. And then I wanted to mention really quickly that there's a companion app that's put out by the developers of iNaturalist, and it's called Seek by iNaturalist. It originally was made for families and children. Things weren't geotagged. It was more about, they gamified citizen science. There were badges and competitions, like how many trees can you get, how many birds, and you can, that type of thing. It became very popular that in the last year or so. They actually made it so that observations that you make with Seek, this other app, you can click a button that says add to iNaturalist. So they've actually connected the two now. And what's really cool about Seek is it tells you all kinds of great information, just like iNaturalist. You can earn badges and challenges and things like that. But you don't even have to take a picture of the item. You literally hold your phone up and it starts IDing it. And so there are these dots that go across the screen here, that the last one is species level. And so if it can get this, so it can get all the way to, sometimes it can get to species, sometimes it can just get to family, but it's really cool. Like the reason I'm showing this to you is, like, let's say you're taking a walk in your neighborhood, you see something, you don't really know what it is. If you have Seek, you can just kind of hold up your phone really quick and see if Seek can maybe help you identify, or at least the area, what type it is. So I think that's kind of cool. And then because of that, for the City Nature Challenge, we have collected a whole bunch of resources about iNaturalist. And so I'm putting this link here because there are all these videos on how to use iNaturalist, the webinars that we did about taking photos for iNaturalist. We did a special session on using Seek and iNaturalist with kids. So there's a recording there. So again, even though this says City Nature Challenge DC, all of the resources on this page are completely applicable for the BioBlitz that we're doing. And then I just wanted to mention, there's one other app that I use a lot, and there's so many apps out there, but if specifically for plants that I found to be really good is called Picture This. And it's been really helpful for me. And then what next? Again, sign up for iNaturalist, join the project. Oh, I put area CNC, sorry about that. So join the Anacostia River Anacostia River Project. Start making those observations, and please take pictures of people and places. We want to see your stuff, but we also want to see on social media, we want to see you guys doing it. So if you're down by the river and you're with your friends and you're making observations of birds or mussels or something like that, let's see some cool pictures of people actually doing citizen science. That would be really exciting for the rest of us to help us promote this event in the future. And there's most of all, just have fun. Just get out there and have a good time. And so that's pretty much it. That's all I have. If anybody has any questions, this is me. Feel free, that's my email. If you want to follow me on iNaturalist, that's my user handle, and you can visit my website.
Yeah, now you can ask directly questions using your audio and video.
Yeah, feel free to, I will stop sharing my screen.
So yeah, tomorrow we will be at Buzzer Point 10 to 1. So if you're interested in learning about aquatic critters, But again, we'll have more of that on Saturday too, actually Friday and Saturday too. And Buzzard Point is very special because of mussels. It's the hotspot of freshwater mussels in the Anacostia River. But then Kingman Island on Friday has had a lot of submerged aquatic grasses in the last few years. So there's like a lot of organisms that depend on that. So yeah, each site is going to have its own unique stuff.
There's so much to cover. So exciting. Well, thank you so much, everyone, for joining us tonight. Again, we did record this, so if you know anybody that could benefit from learning about iNaturalist, Jorge will be sending out a link, I think we'll be sharing the link to this presentation. So I hope to see, I'm going to be out, I guess on Saturday, I'll be at Kenilworth Aquatic Gardens with some other folks. So I'm looking forward to seeing people live in person, yay, so that we can all nature nerd out together. Yeah, I'm looking forward to seeing what we find. And one of these days I need to get on one of the mussel surveys with you guys, you and Laura, you guys. So that looks really fun.
Absolutely, yes. So happy BioBlitzing. Let's see who, normally that's a tradition. Let's see who's the first, who makes the first observation after midnight.
Oh man, I wish I lived near there because, you know, for the City Nature Challenge, I'm always out there at midnight trying to make the
first observation will be Carrie. She'll go out and get some spider.
It's true.
Yeah, it's not gonna be me because my 2-year-old daughter's gonna wake me up at 5:30.
Well, have a great time, everybody, and we look forward. And one thing I wanted to mention, there could be, just kind of stay tuned. We might have a virtual ID night where we all get on a Zoom in the coming days, weeks, whatever. It just hasn't been organized, but it could be where we all get on and we help each other make identifications. So we've been doing that monthly. There's a group of people doing that monthly each month on different types of things. And if we can get it together to do this for the BioBlitz, I think that would be a fun activity too.
So yeah, we will announce the results on the week after. Why is that? Because, you know, it takes some time for people to upload and make observations. Again, you can, although we recommend to upload things as soon as possible, you can upload them later because they are, you know, tagged the date and time. They even if you upload them on the Monday after, if they're tagged for the right time, you know, and you did that during the weekend, that's going to be okay. But again, we recommend to do it as soon as possible so that we get some exciting numbers.
Absolutely. Absolutely. All right, guys.
All right.
All
Anna,
right.
thank you so much. This was very helpful, and hope to see you BioBlitzing tomorrow.
Yes. Hope to see some people this week. And yeah, if you were on this session and you come on Saturday, definitely come up to me and be like, I was on the session, so I can say hi to you. Yeah. Great. Take care, guys.
Bye.
Bye.
Thanks. Bye-bye.
Bye-bye.
Hi, good to see everyone. Sorry I was late.
No worries. Take care.
No worries.
Okay, see you this weekend. All right,
Okay.
see you tomorrow.
See you.